Thank you, Tom. At this time, I'd like to start introducing our presenting companies. Our first presenter this morning will be Certerra, Inc. Certerra is a contract research organization with specialty in central nervous system based on single neuron level monitoring of mammalian brains. Presenting this morning for Certerra is Pavel Ostin.
So thank you, Neil, for including us today this morning, and thank everybody for coming. As Neil mentioned, we are a contract research organization. We work with small and large pharmaceutical companies, and our specialty is screening animal models at the level of single neuron resolution. We hope that our technology will improve the really important step of transition between preclinical drug screening and taking a drug into the clinical trials. So how does our technology works? We have transgenic animals that translate, when the animal is given a drug, that translate the drug-evolved brain activation into labeling individual cells in the entire brain, via green fluorescent protein. We have automated microscopy that images those brains as a serial sections of 280 optical sections from the beginning of the brain to the end of the brain. And then we have computational methods that detect all the activated brains, detect all the activated cells, register the cells onto an anatomical atlas, and then we run statistical assays that compare animals that were treated with a saline to animals that were treated with a drug. And that results in what we call a pharmacomap analysis, which is a number of brain regions that were activated by the drug, and number of cells within those regions that were activated. This pharmacomap technology can be thought of as in translating the drug-evoked activation into a computer code. So we count the cells that are activated as ones, and the cells that were not activated as zeros. So you can think about this technology as a brain chip for drug-evoked activation. And we can calibrate this brain chip by using drugs that we know how they work in the clinics to predict the effects of the drugs that are in the preclinical drug discovery. So we first start with taking drugs that are used in the market and for which we know the clinical indications and we know the side effects, and we screen those drugs by our technology. We generate these, what we call an animal to human or A2H database, and then links the patterns in the mouse brain, and then links the clinical effects of those drugs. We then take a new drug, which is in preclinical screening, and we screen it with the same methodology. We compare the pharmacomap pattern of the new drug to the database that has the drugs that are known from the clinics. And we predict based on the overlap between the positive areas that would be the indications of the drugs. We predict what the drug will do in terms of its indications. And based on the overlap in the areas where the previous drugs highlight and side effects, we would predict the possible side effects and adverse effects of these drugs. So the technology is applicable to all drugs in preclinical screening. It's applicable to brain disorders, because that's what we screen in this technology. But it's also applicable to drugs that are not targeted for brain, because you want to know if you're developing a drug, if it potentially will have a side effect once you take it into the clinical trials that would be based on brain effect that would not be predicted initially. Our main focus now is on disorders such as schizophrenia, depression, Alzheimer's, and dementia, so on cognitive disorders. Currently, if we look at what is our immediate market, currently there are 97 companies that have 162 new drugs in early clinical trials in the phase 0 to phase 2. Our market out for these 162 drugs would be about 20 drugs that are at the level of animal screening, so this comes about to 3,000 compounds of an immediate market that we would like to address. 3,000 compounds based on our current pricing is about $180 million market. Based on our projected growth, we would like to have about 2.5%, so very modest penetration initially, into achieved in the 2015. So there will be about generating revenues about $4.5 million. And then on increasing growth, about 10% penetration in 2016, or revenues of about $18 million. Our success so far, we have raised a seed funding of about $1 million, or $1 million from Houseman Partners. We have filed a patent application to protect the pharmacomet technology. In addition to the patent application, the know-how and the database that we're generating is making additional barriers to entry for our competition. In the 2012 to 2013 that we've been in operation, we have generated an initial revenues of about $700,000, but this was really based on a pilot project that we had as we were developing the technology. And we have received an SBIR grant of $2.1 million from the government, which is for generating a database of mental health medications. So this is the part when I was showing you, and then generating a database of animal to human that has drugs that we know how they work in the clinics. Our competition, so the most sort of easiest to compare our competitions are small animal imaging companies that use an MRI and fMRI imaging technologies. However, it's not really an comparable technology. What these companies, mainly what they do is an imaging in the periphery for cancer. There's only a small market for them in terms of imaging in the brain, and that's because the resolution of MRI and fMRI imaging is much lower than what is needed for really mapping and drug-evoke activation. So it's looking at a blood flow instead of looking at an activated cells, and it has also a lower spatial resolution. I have put one and a successful company that was based on assay, and that's an Illumina. So in the late 1990s, there was a lot of sequencing technology. It started as an assay technologies, and you can see that today the market cap of Illumina is a $17.9 billion. So even an assay company can generate a substantially large value if the technology is disruptive and transformative, which is what we believe our technology can do for drug screening. And finally, I'd like to mention our growth strategy. So we're seeking about $5 to $6 million in funding. Most of the funding will be on capital expenditure. So we have currently be operating with one instrument. We plan to add 9 additional instruments. So essentially having 10 times the throughput that we have currently. We are actively searching for a CEO. We hope to add about 10 to 15 scientists to the team that we have currently, which is 3 scientists. As of now, we are cash flow neutral, and based on what I was saying in terms of the penetration of the market, we hope to have revenues of $4.5 million and $18 million in the 2015 and 2016. Thank you.
All right, so we have time for 1 or 2 questions from the audience. Any questions?
How standardizable is that? How much of it is get the data, press a button, and funnel it through?
It's almost entirely you get the data and funnel it through. So the only handling of the animal is the drug delivery. After that, the imaging, the computational detection, that's all standardized. And we are going to go to the science details of that. The measuring and the standardization is due to the statistical significance. The activation regions that we have come to 10 to the minus 10 to the minus 15s statistical significance. So it's comparable to sort of what you would see in human genomic screening. Highly standardized. We're talking exactly about this project with Eli Lilly right now. What we have done for the phase 1 part of the NIH grant is to take 20 drugs, 2 per class, screen them at low level of 5 animals per drug, and show with the PCA that we can get an essentially signature for all of the classes. And there was a 99.2% probability of identification of the class based on the pharmacometrics. But we need to have the drug that fails to sort of fill in the gap of what we would be looking for. Thank you very much.
Well, thank you. That was so great on so many levels. Most importantly, to me at least, Paolo promised me before he started that he was going to finish right on time, and he actually finished spot on to the second. So with that as a precedent for the remainder of our companies this morning, let's, this is still you, right? Okay, let's move on to our next presenting company, which is Autoimmune Therapeutics. Autoimmune Therapeutics develops orally available drug candidates to treat autoimmune disease with initial focus on rheumatoid arthritis based on proprietary small molecule scaffolds and pharmaceutical leads that inhibit macrophage migration inhibitory factor. I cannot say that again quickly. Presenting this morning is Tom Coleman.
Thank you. Thank you again to the group for having us, and in particular to Corey Bazella, who helped me was my coach, if you will. Autoimmune Therapeutics.